So as it was mentioned before, the objective of our research was to create a pipeline of computational analysis from DNA fingerprints, which are sequences of DNA broken into unique segments. So before we begin, we want to give a basic understanding of microbiology and why we chose the microbes we chose. So to start, probiotics are the live beneficial bacteria that live inside of your body. Their job is to keep your body healthy and fight off any bad bacteria that may come in. They make up a larger picture called your microbiota, which is involved in much of the physiology of your body. So not only are these probiotics found within your body, but also in the foods we eat, specifically in fermented foods. So lactic acid bacteria is a specific bacteria found within fermented foods. And for the purpose of our research, we chose to study kefir and buttermilk. Kefir has bacteria listed on the ingredient list. So once we got our result, we were able to compare it back to see if it matched any of the ingredients. Buttermilk was more of a challenge for us because it had no bacteria listed on the ingredients. So we wanted to see if we were still able to identify a lactic acid bacteria. All bacteria contains a 16S region in the ribosomal RNA. So this is a component of the ribosome, which is important in producing protein. What makes this important for our research is that it is a highly conserved region, so it shares some of the same characteristics and structure between all bacteria. However, there are also variable regions that allow us to differentiate each bacteria from one another. So the typical method for identifying DNA sequencing is very inconvenient for most labs. DNA sequencing machines can cost $20,000, so most labs don't have one or can't get access to one because even sending in samples can be expensive. So these inconvenient methods leads us into the goal of our research. Probiotics are beneficial to your body and in your diet. So we wanted to create a cost and time effective method of identifying lactic acid bacteria so that people have a better understanding of what they're putting into their body on a daily basis. So we did this by comparing the 16S sequence of the experimental biology team's data to the 16S sequences of the computer science team's online database, and this left us with a computational modeling method which took the 16S sequence of lactic acid bacteria and was able to identify the genus and even in some cases the species of this bacteria.